a graph that would be obtained when assessing antibiotic potency using agar diffusion. And that was an example where two standards are used. And you can use that graph to calculate the potency. Okay. So there are several considerations that are needed when actually carrying out the assay, and this involves the results that you obtain on the agar plate. Some of these considerations are: one, zones which are not perfectly circular, because you are dealing with diameters, so you need good circular shapes. Those ones which are not very perfectly circular can be ignored. However, the different dimensions may be measured. The smallest and the largest dimensions is maybe a good thing to have that record. Some of the reasons why the zones may not be circular are: one, if the wells themselves where the antibiotic was placed was not circular from the beginning, so the shape will also not be circular. The other reason might be if the agar is not dried evenly. So in that case, the moisture content of different points at different points of the agar is different, and therefore the diffusion of the drug will be different. And the other part is not being very careful when when filling or when putting the antibiotic. So if the solution is not put carefully, then we may have those irregular shapes. During measurement, the measurement can be taken directly using calipers, but the most precise and accurate method would be to project those shapes on a screen and taking measurements from there. So that's kind of a magnification activity, which would make measurement much easier and more accurate. In some cases, there is possibility of automation, and this uses photocells. So where that kind of an instrument is available, then that would be more accurate, precise, and it is able to produce any other statistics which are required. There are several factors which affect zone diameters. So it's important that certain controls are put in place so that there is reproducibility. One of them is presence of other solids like buffers or sugars, and this will depend on the product which is being analyzed. So if it's a syrup, there will be quite some sugar. If they are tablets, then you get different types of excipients, etc. So those other products, those other compounds present may affect the diffusion of the drug and therefore the size of the inhibition zone. The other one is gel strength, which is basically the type of gel and how it was prepared. These diameters may not affect accuracy from day to day. However, the precision might be affected. So as you remember when we were discussing some of these terms in the other unit, it makes sense that accuracy you may get the same level of accuracy, but the zone diameters will be different. They may not be very reproducible because of some of these factors. There are newer variations to the agar diffusion assay, whereby instead of having the drug put in a well, it is actually placed using a disc. So you have adsorbent discs containing different drugs, which are then placed on the agar. So this would be more reproducible because the discs are produced, they are available commercially, so they are better standardized. Okay, so that is the other type of assay that can be done, test antibiotic potency, and they are called turbidimetric assays. This simply means that instead of having solid medium, you now have a solution or a broth as the test medium. It follows the same protocols whereby several concentrations for the test and standards are applied, and then the growth of microorganism is determined by measuring the change in turbidity of the broth. So this can be achieved by use of nephelometry or spectrophotometry. So this particular method is much rare because of its disadvantages, which include lower precision. So it's harder to obtain similar results with regard to the measurements. So it's less common. The agar method is more preferred. To improve on the outcomes, these tubes are incubated in a random manner, and this randomization is achieved by use of Latin square. That's the same as we saw in the agar diffusion method. For this case, because of the volumes that are involved, the tubes need to be shaken regularly to improve aeration. So this ensures that there is uniform growth in the tube, not only in some parts of the tube where the oxygen might be much more available. So shaking, a shaker would be required to do this practical, requiring only about 3 to 4 hours. And because of the rapid growth of microorganisms, they tend to be more sensitive. So for substances which may not be very potent, maybe during drug discovery, you're trying to see whether an extract contains some antibacterial activity, that this would be a good way to detect such kind of activities. So as a result of what we have discussed, that there is a lower precision, then we find that the dose response curves are more variable and the plots are usually non-linear. So this may not be a very good way of quantification, but it's a good way of detecting some activity in poorly in compounds that may have poor activity. Yeah. So another application of microbiological assays is in the determination of minimum inhibitory concentration. So by definition, minimum inhibitory concentration is the lowest concentration of an antimicrobial, which is able to inhibit growth of a test organism. So the MIC is dependent on the microorganism that is used for testing. It is the fundamental measure of antimicrobial activity or potency for compounds which are used as antiseptics, infectants, and preservatives. Most often, it is applied to raw materials rather than the final products. So before maybe a product is formulated, then this is an important test to be performed. For final products, they are more likely to be tested using other tests, such as preservative efficacy, challenge tests, etc. The minimum inhibitory concentration is an absolute value. So it is reported as a certain concentration, maybe in milligrams or units per ml. So there is no relativity, like in other cases where we have the value being obtained as relative to maybe another standard. So this is an absolute value, and therefore, if proper controls are not implemented, then the result could could give significant errors. So it's critical that certain precautions are adhered to. There are different methods which are used to carry out minimum inhibitory concentration tests. They may use test tubes or micro titer plates. So these micro titer plates, we have seen such examples. And these micro titer plates are more efficient because they hold small volumes of test solutions. So the controls which are needed are intended to show viability. So this is just to confirm that the microorganism being used for testing are actually viable, so that you don't report that the drug was effective, yet the organisms that were used to are actually not viable from the beginning. They are also intended to show that contamination did not occur during the testing. So those are the different controls which are needed during the actual testing. Besides that, all other solutions which are used must be sterilized. So only the test organism solution is not sterilized. All the other products, all the other solutions, all the tubes and plates that are used must be sterile. So the advantage that is obtained through this test is that it is possible to detect microorganisms that contaminate, and in case the growth is not very vibrant, it is also possible to observe. Another practical aspect of the carrying out the tests is that the concentrations, subsequent concentrations, are usually doubled. So that if you start with 1 microgram per ml, you go to 2 microgram, then 4, then 8, etc. So that way, you're able to go over a wider range of concentrations using a very fewer number of tests to run. Another set of tests are known as the preservative efficacy tests. So as the name suggests, they are intended to test how well a preservative works in real life. And we know preservatives are substances which are added to a product to ensure that growth of microorganisms does not occur. So they are also known as challenge tests. It simply means that a final product is intentionally contaminated with a defined number of microorganisms to check the growth, or to check how well that substance prevents the growth of that microorganism. Microbiological assays are required to detect or to assess preservative efficacy, because chemical tests cannot predict the effect of other constituents as well as the container on the effectiveness of a preservative. So we know that certain types of materials for containers can actually absorb preservatives, which are usually added in low concentrations. So if a container is absorbing a preservative, that means then the expected concentration in the product will be much lower, which may result in low effectiveness of the preservative. So the effectiveness of a preservative would then be best tested using a biological test. Preservatives will most likely be present in certain formulations, such as multi-dose injections, eye drops, a majority of oral mixtures, those that don't contain highly concentrated sugar solution, so like Linctusses, is also going to be present in creams and lotions. There are, however, other products, especially those that do not contain moisture, or what we call anhydrous products, such as ointments or single dose injections or large volume injections like infusions. So those are other products which may not have a preservative in them. So another question we may want to answer is whether an antibacterial cream requires a preservative. And the answer to that would be yes, because not all antimicrobial are self-sterilizing. So if you have an antibacterial cream, they may not be effective against fungal contamination and growth or spoilage. So it's important to have a preservative which then prevents the growth of fungi or yeasts, for example. So it's important to note that not all antimicrobial agents will be self-sterilizing or will not require preservatives. How is it performed? How are the tests performed? It requires the use of different containers, whereby in each of the container, a known quantity of a microorganism is added. So we're talking of the range of 10 to the power 6, 10 to the power 5 to 10 to the power 6 per ml of microorganisms being added to each container. And this large number of microorganisms is good because the eventual test requires dilution of 10 to the power 3 to 10 to the power 4 factor. This dilution is especially critical because if the preservative remains concentrated during assessment of how effective it was, then the carryover effect of its power will be carried over. Okay? So it's important to have this large number of microorganisms so that we can have the desired number of microorganisms on the plate during counting. In addition to that, or before we go there, we can say that this is followed by drawing samples from each of the containers and then assessing the proportion of microorganisms that survive. So during this assessment, we may need to inactivate the preservative. So we have noted that preservatives are added in low concentration to products. We are also carrying out quite a bit of dilution, but in some cases, inactivation may be required. For example, if you are to incubate a series after a series of samples, and then you observed no growth, you'd not be certain that the microorganism or the preservative has killed all the tests or the microorganisms. So you may need now to do a slightly less diluted solution. So because we were diluting 10 to the power 3, a factor of 1,000, you may want maybe less. Lysine for aldehyde preservatives. We have thioglycolate or cysteine for heavy metal-based preservatives, and mixtures of lecithin and polysorbate 80, with or without a product known as Lubro, L-U-B-R-O-L-W, for quaternary ammonium compounds, chlorhexidine, and parabens. So preservative inactivation, we have noted, is required, especially where there is no growth after that dilution, and then we have the chemicals that can be used. Besides that, we can physically trap the microorganism by filtration and then culturing. So that is still another alternative to use of chemicals. So we can physically isolate microorganisms using filtration and then taking whatever is filtered and culturing. So there are certain microorganisms which are recommended to test preservative efficacy, and these are bacteria, which include Staphylococcus aureus, Pseudomonas aeruginosa, and E. coli. For fungus, Candida albicans, Aspergillus niger, and in some cases, Zygosaccharomyces ruxi. So those are the recommended microorganisms. It depends on which pharmacopoeia you are looking at, and then you get different microorganisms. There are, however, some which are common in all of them. Any questions so far? Okay, there is no question. We go to disinfectant evaluation. So microbial biological assays are also used to assess how disinfectants are working, or the potency of disinfectants. So various tests are employed to assess disinfectants, but they can be classified according to the test design. There are other ways in which they are classified, but we will go by classification by test design. So the first one is the suspension test, and this involves the addition of a defined concentration of test organism at a specified temperature. So temperature is important, followed by assessing the viability of the inoculum after a suitable time interval. So in the suspension test, a defined concentration of test organism is added at a specified temperature to the solution, and this is then followed by assessment of viability after a suitable time interval. There are certain things that we need to note when it comes to disinfectants and the evaluation of their effectiveness. The first one is that in normal usage of disinfectants, there is quite a bit of organic materials present, which we can call organic dirt. So there is quite a bit of organic material on the surfaces, on where they are actually being used. So the tests try to simulate these actual use conditions, and some yeast and albumin or other organic material is added to these tests. So this tries to simulate what would happen when the disinfectant is applied to a surface. The other thing to note is that the disinfectant must be inactivated. In other words, when the sample is drawn from to test for viability, then the effect of the disinfectant must be stopped so that we are able to assess how effective it was within that time span where you are required to do the test. So this therefore makes it's more meaningful. It's important to carry out this inactivation. The other thing to note is that if you check some older books and the newer process, there are some changes in that older methods used were testing the viability after hours or minutes after the disinfectant has been added. But it was also testing viability by incubating in broth. So even if there were just a few microorganisms that survived within the first few minutes or hours, then these resistant microorganisms may actually grow in the broth. So this would make a disinfectant which is effective be discarded as not helpful. However, it will have reduced the biological load by a significant level. So current methods test viability after 28 days. So that's the difference between the old methods and the newer methods, whereby viability after 28 days is determined. And because the tests involve counting the viable microbes, then there is what is known as the criteria for determining the performance of the disinfectant. So they use what is known as a 5-5-5 testing principle. This refers to the use of 5 different microorganism species which are added separately to different tests. And these microorganisms are then exposed to the disinfectant for 5 minutes. So within this time span, they are expected to have reduced the viable load, the viable microbial load by 5 log reductions. So that means if you started with a certain number, then you would expect 10 to power 5 4 in viable counts per ml. So that's what the 5-5-5 refers to. 5 different microorganism species exposed for 5 minutes, and the expected result is a reduction, a 10 to power 5 reduction in viable counts per ml. The other types of tests that we just mentioned are capacity tests, and these are intended to measure the extent to which a disinfectant can start addition of microorganisms. So this is also a normal usage process, because we know that once you apply the disinfectant in a certain place, then additional microorganisms can still land there. So how well is the disinfectant able to hold that? The other test is a carrier test, and this uses a microorganism loaded or dried on a carrier. And then finally, in-use tests, and these tests are designed to simulate actual conditions of use as much as practically possible. So those are the different types of evaluating or different methods of evaluating disinfectants. Any question? Okay, so if there are no questions, that brings us to the end of that series of discussions of microbiological assays. And we can then start off on immunoassays. So by definition, immunoassays are quantitative assays which depend on a reaction between an antigen and an antibody. So those tests which rely on this particular reaction are known as immunoassays. And we know that these are specific interactions. We mentioned them. We mentioned antigen antibody when we were talking about affinity chromatography. So we can now also utilize antigen antibody reactions to determine the quantities of either, depending on what are we interested in. Basically, immunoassays are based on a competitive binding reaction between a fixed amount of labeled form of an analyte and a variable amount of an unlabeled sample analyte. So in this case, we can say you have a standard, something which is already known, and how much it is. It is mixed with an unknown, so which is basically the sample. And this is supposed to compete for a limited number of binding sites. So these binding sites are actually highly specific anti-analyte antibodies. So when these reagents are mixed and incubated, the analyte will get bound to the antibody, forming an immune complex, which is an antigen antibody complex. There are two ways in which, or there are two different methods. In one of the methods, the complex may be separated from the non-complexed reagents. In other methods, the measurement of whatever is bound or whatever is not bound is done within the mixture. So in some cases, there is separation of the complex from the unbound, and in other methods, the two may not be separated. So to determine how or what proportion of the reagents interacted different types of labels are used. And these labels are actually ways in which we can determine what proportion formed the complex and which proportion remained free. So these labels may include measurement of radiation, or that is a measurement of fluorescence or enzyme activity. So this radiation is actually radioactivity. So there are different ways or different labels that can be used to measure what proportion changed form. After this measurement, then it is possible to produce a standard curve which represents the measured signal as a function of the concentration of the unlabeled analyte. And it is from this calibration curve that we are then able to determine the concentration of an unknown. The immunoassays, as we have outlined, then requires the following materials. It requires antibodies. There are different types of antibodies. You see them, what types. We also require signal generating labels. What we are saying that you can measure radioactivity, fluorescence, or enzyme activity. And then in those cases where you need to separate, you need effective means of separating the complex from the free forms. And those are the separation matrices. So the simplest outline of an immunoassay follows the following outline. The reagents are mixed and incubated. So these reagents would include a fixed amount of the analyte, antibody sites, anti-analyte antibody sites, a fixed amount of labeled analyte, and a variable amount of the unknown or the standard. In some methods, there is separation of the antigen-antibody complex from the free antigen and antibodies, and this can be achieved by physical or chemical means. The third step is to measure the activity of the label, and this can be done either on the complex or the free portion. The fourth step is to prepare a standard curve and use the same to determine the concentration of an unknown. So let's look at the details of these reagents. We start by looking at the antibodies. So the antibodies determine the success of an immunoassay. This is obvious because they are the key that will interact with the unknown. So there are two types of antibodies, monoclonal and polyclonal. And this is more to do with the source. Is it from a single type of cell, or from different types of cells. So those that come from a single cell, a single type of cell, they are monoclonal. And those that come from different types of immune cells are polyclonal. So in most cases, monoclonal antibodies are more advantageous because they are produced by selecting cells that produce antibodies with high affinity and high selectivity. So they would be more advantageous in a pharmaceutical analytical process. However, there are still successful methods that have used polyclonal antibodies. The other bit of the reagents are the signal generating labels, and we will look at them in more detail later. So we'll just mention them. We can have radioactive atoms, enzymes, fluorescent probes, chemiluminescent substances, metals and chelates, and liposomes. For radioactive atoms, this can include iodine-125, tritium, that is H3, and carbon-14. So radio immunoassays were the initial tests that were developed. They are quite sensitive and very precise, but they have certain disadvantages, which include the health hazard. They also required special attention for handling, so they require more training, more experience, etc. And they also tend to have a short half-life of the isotope. In addition to the requirement of having expensive instrumentation for counting radioactivity. So they have those advantages and disadvantages, that they are sensitive and precise, but they have those quite serious disadvantages. And these disadvantages therefore led to the development of the other types of labels that could be used to detect these changes. By far, the most common type of label used is the enzyme for the pharmaceutical compounds. This is because they offer a chance to magnify or to amplify the observed signal. So we know that enzymes catalyze reactions and they are not used during the process. So as long as you have a bit of the enzyme, then the reactions will proceed. And this increases the signal or amplifies the signal. So this tends to improve sensitivity of the tests. The next bit of the materials, or the components of the immunoassays, are the separation matrices. include charcoal, polyethylene glycol, second antibodies, microbeads, and the most common and most useful of them all is the 96 microwell plate. So these plates also allow some bit of automation, as we have mentioned previously. The use of these microwell plates facilitates washing steps where washing steps are required. They are also very useful when pipetting reagents. So they are micro pipettes which can be used to add the solutions to these wells. And as we have mentioned, semi-automation is also possible. If we look at the different methodologies of immunoassays, they can be classified based on whether there is a washing step or there is no washing step. So in those methods where there is a washing step, then that is classified as a heterogeneous method. And where there is no washing step, where everything is in one pot, then that method is described as being homogeneous. It can also be classified based on the design. If the method is based on competition, either of limited number of binding sites or limited number of analyte, then that is a competitive design. And where there is no competition, then that method is known as non-competitive design. In majority of competitive designs, then we can have either the antigen fixed or the antibody fixed, such that the initial step involves capturing of either the antigen or the antibody respectively. So the competitive design can either be a capture format or an antibody capture format. We'll see these details very shortly. So that is an outline of an antigen capture immunoassay. And we can see that the first part there, you have the antibody, which is coated onto the solid support, which could be the 96 well plate surface. The second step is the addition of analyte and labeled analyte. So this if I can get. Okay, so the diamond shapes are the analyte, and the one with an attached L is the labeled analyte. So once that mixture is loaded onto the coated plate, they will compete for the limited number of available binding sites. So depending on relative concentrations, then they'll bind accordingly. Okay, so whichever is more concentrated, either the labeled or the unlabeled, then you get a similar proportion being bound to the well. This is followed by washing. So whatever is not bound to the fixed antibodies will be washed off. And you can see that in proportion, whatever was not bound will be washed off. Looks like in this case we had more of the sample and the unlabeled, which was more concentrated, then a higher proportion of it will be bound to the bound antibodies. This is followed by assessing the activity of the enzyme. Yes, yes, Walter.
Are we washing both the antibodies and the antigens?
Yes, what in this particular setup, whatever is being washed off is, you can see we have a mixture of, need to get some different colors. Let me see where to get those. Okay, you can see the key down here. You have the analyte, and then you have the analyte with the label. So the label is the enzyme. Okay, so the first step, we are adding this step here. We are adding a mixture of these two, the analyte and the labeled analyte. For the labeled analyte, we know how much is present. For the analyte, we don't know how much is present. So depending on the relative proportions, then they will bind by those relative positions to the bound antibody. Okay, so this antibody which is on the wall of the plate, they will compete for those antibodies, and they will bind by the same proportions they were in the mixture. Is the same proportion you would expect on the bound antibodies. So what is being washed off is whatever is not bound to the bound antibodies. Whatever is not fixed is the one that is going to be washed off in that step. Is that clear?
Yeah. So I think now my question was, to be more specific, is whatever is unbound, could it contain both the analyte and the labeled analyte?
Yes, it is going to contain the analyte, some of the analyte, and some of the labeled analyte. So whatever free, in other words, whatever does not bind to the fixed antibody will be washed off. It could be analyte or labeled analyte. So the important thing to note is that the same concentration that is the proportion, they bind relative to their proportions. So if you have 20% of the analyte, and 80% of the analyte. So 20% of the analyte is going to be bound on the fixed antibodies, and 80% will remain in the washing. 80% of whatever is bound will be labeled, and similar concentration in the washing. So in other words, whatever is bound, the proportions in the bound and the unbound fractions is the same for the different species. I don't know if it is making sense.
Yes.
Yeah, similar proportions. So it's about proportions. It's not about absolute numbers. So we are saying that the binding sites are limited. So they are much fewer compared to the added analyte and labeled analyte. So the binding will be the same as in the same proportions as in the original mixture. So they compete, and because they are not very different, you would expect similar proportions. So after washing, then you are left with the bound, the complexes that will form will either be the complex between antibody and analyte, or between antibody and the label. So the next step, yeah. So the next step is the addition of the substrate. Okay. So we are talking of depending on the label that we are using. If it is radioactivity, we just measure radioactivity. If it is an enzyme, then we will be adding the substrate. So and so on and so forth. So the next step would be reading the label activity. So this might incorporate the addition of the substrate for an enzyme, for example. Or it could be simply taking the tube and putting it in the radioactivity counter, or taking the tube and taking it into a spectrophotometer. So depending on whatever label we have, then we read activity. So the signal which is obtained relative to the concentration of the analyte will be inverse proportionality. So you can see that I need to be able to okay. So as the concentration of the sample is increasing, then the activity will decrease, because more of the available sites will have been bound to the analyte. So as the concentration of the analyte increases, then the proportion of the labeled analyte that binds decreases, and therefore we get that drop of activity or signal as the concentration of the analyte increases. That making sense? Even the ones just listening, you can write something on the chat. I'll be able to see. Okay. So then we can go to the next one, which is antibody capture. So in this case, whatever is fixed is the antigen, and then we are adding a fixed amount of labeled antibody combined with the analyte. So the antibody will compete either to bind the unbound analyte, which is from the sample, or it will bind to the bound analyte, which is fixed on the slide, okay, or which is fixed on the plate. After washing, then you'll have some of the labeled antibodies being washed off together with some molecules of the analyte. But the important thing is that the bound enzymes or the labeled enzymes, the labeled antibodies that you bind to the bound antigens will be the same proportion as the proportions of labeled and the sample, all of the fixed and the free sample or analytes. So the next step is follow, just like the other method, is reading the label activity. And the proportions or the response will decrease with increasing concentration of the analyte. Because in that case, the higher proportion of labeled antibodies will be washed off because they have bound to the sample molecules. No questions? Okay. So immunoassays that are of pharmaceutical applications can be classified based on the type of labels that are used. And examples include radioimmunoassays, as we have outlined, enzyme immunoassays, colorimetric immunoassays, chemiluminescence immunoassay, and liposome immunoassay. So we look at each of them. So we have radioimmunoassay. So radioimmunoassay methods have been used for very many compounds, and especially where biological fluids are involved. So we know that biological fluids are quite complex. The concentrations of these substances are also very small. So you need a sensitive method that is also not affected much by these complex matrices. So radioimmunoassays were very useful. Most of the current methods, radioimmunoassay methods, are automated, and the separation is assisted by using antibodies which are bound to a solid phase matrix, so that like the 96 well plate. So what we have is just a very small example of drugs which have been assayed by radioimmunoassay methods. The samples on which this is applied, and then the sensitivity. And you can see we are talking of very small concentrations where this method has been used. So we have mentioned that this method is quite sensitive, has high precision, so it is very reproducible. And it is only close in regard to those two aspects to GCMS, gas chromatography with a mass spec detector. So in terms of sensitivity and precision, radioimmunoassay is up there. Then we have enzyme immunoassay. So in this case, the label is an enzyme substrate pair. So it utilizes the catalytic properties of enzymes to detect and quantify immunologic reactions. So the basic approach for use of an enzyme as an immunoassay label will only be appreciated by understanding that the enzyme converts a substance, a compound which we call the substrate, into a product. When these two, the substrate and the product, should have different properties that we are then able to quantify. For example, you can have a product which absorbs poorly at one wavelength, while the substrate which absorbs well at a given wavelength, where the substrate does not absorb significantly. If, for example, you are talking of a spectrophotometric assessment or measurement. So this way, you are able to measure the intensity of absorption as a function of the product formed. This, the product, the intensity or the rate of formation of the product will depend on the availability of the catalytic sites. So you need to have the reaction between, or the binding between, the antigen and the antibody affecting the activity of the enzyme. So we shall see the different ways in which that is achieved. So enzyme immunoassays can take different forms. We can have heterogeneous We shall see an example of heterogeneous enzyme immunoassay. We shall also see an example of a homogeneous enzyme immunoassay. They could also be competitive or non-competitive, and the formats could be antibody or antigen capture. We have listed a series, a sample of enzyme substrate pair that have been used. We have alkaline phosphatase, which is the enzyme with either P-para, that's para-nitrophenyl phosphate, or streptavidin as the substrate. We have an example of beta-D-galactosidase with nitrophenyl galactopyranoside as the substrate. We have horseradish peroxidase with 3,3,5,5-tetramethylbenzidine as the substrate, and glucose 6-phosphate dehydrogenase with NAD as a substrate. So these ones can be used with spectrophotometric measurements because the products absorb at different wavelengths. And we have mentioned that there are different methodologies, designs, and formats for enzyme immunoassays. So we'll just look at two examples. Starting with this one known as ELISA. ELISA is a short form. It's for enzyme-linked immunosorbent assay. It is a heterogeneous enzyme immunoassay technique. So in this type of assay, one of the reaction components is non-specifically adsorbed or covalently bound to the surface of a solid phase, like the microtiter plate, a magnetic particle, or a plastic bead. And this is aimed at achieving or facilitating separation of the bound and the free labeled reactants. Which are the steps which are involved? So the first step, just like we observed or we have mentioned, is coating, whether the antibody or the antigen on the plate, and then washing. So each of these additions is followed by washing. The second step is to block, and this is because some antibodies may have non-specific binding sites which are blocked by addition of proteins such as albumin. Okay? Bovine serum albumin is added. So this blocks non-specific sites. And then this is followed by washing. The third step is addition of the sample, followed by washing. So the sample will add the analyte, which then binds to the specific binding sites of the antibody. So what we have, this is the coating antibody. This is the non-specific binding, which is blocked. And this would be our analyte molecule. So addition of the analyte is followed by addition of the specific antibody, which binds to a different epitope of the analyte. So there are some molecules which can be recognized on two different sites by different antibodies. So this a site where an antibody is able to recognize is known as an epitope. So such a molecule would be capable of being identified or binding to two different antibodies. So in this case, we have such an analyte. So by addition of the second specific antibody, then provides additional identification of that analyte. The fourth step is that addition of the second antibody, specific antibody, and this is followed by addition of the labeled antibody. So why they go to this other antibody is because you can produce specific antibodies against the analyte, which can then be quantified by using commercially available labels. So the labels are able to be obtained commercially, and that reduces expense. So rather than directly trying to produce antibodies that are labeled for your own experiment, then you can obtain commercially available labels, which makes your work easier. So the final step obviously involves the addition of the substrate and measurement of whatever change is taking place. So the most common being spectrophotometric, where there is a change in absorbance at a given wavelength as the product is formed. So the rate of this change or the quantity of this change within a specific time then is an indicator of the activity or of the concentration of the substance. So if you read some material, you'll see that ELISA can be classified into direct, indirect, sandwich, or competitive. So all these have their specific applications. The one we have outlined here is a sandwich ELISA. As you can see, the substrate or the analyte is sandwiched between two specific antibodies. Another example of enzyme immunoassay is a homogeneous type, which is known as EMIT, which means enzyme multiplied immunoassay technique. So this is a homogeneous assay where everything is mixed in a single pot. And it is a competitive type of assay. So the steps are the enzyme, which is labeled with the antigen, which is the analyte. So we have that left bit there is the enzyme with the analyte attached close to the active site. So to this enzyme is added the analyte. So this is the analyte. Okay, so we have free analyte from the sample, and we have the enzyme with the bound analyte. This is followed by addition of the antibody. So once the antibody is added, it can bind to either the analyte bound to the enzyme, in which case that binding blocks the active site of that particular enzyme molecule, or it can bind to the free sample analyte. So this one is bound to the free sample analyte. This is bound to the analyte bound to the enzyme, which blocks the active site of the enzyme. Okay. So the final step is addition of the substrate, and the substrate will only be acted upon by the free complex or the free labeled enzyme. So the one that is bound to the antibody will not be active, only the unbound or the uncomplexed enzyme will act on the substrate. So the intensity of the change in absorbance at a given wavelength then is a measure of enzyme activity, which is the proxy for the concentration. So as the concentration of the analyte increases, it means that a higher percentage of the added antibody will be bound to the analyte molecules, the free sample analyte molecules, rather than the ones bound to the enzyme, and therefore enzyme activity will increase. So there is direct proportionality of activity. As the concentration of the analyte increases, the activity of the enzyme increases. Yeah. That makes sense? Any question on that? Okay, it's no question. So we go to fluoro immunoassay. So in this case, we have the label being a fluorophore. So we are measuring the amount of light emitted at a given wavelength after irradiating the substance with a different wavelength. Yes, Walter?
I was requesting if you can use an example of a drug in the market as you are explaining, so that it can be kind of easy to do a relation between.
Okay. What I can do, there is a link I will send where you can see some of these methods being applied. So some of them are reported in journals. Some of them have been synthesized into a document. So it's a whole range of drugs. So it may not be very useful to give specific examples. However, like in the case of EMIT, this is used for monitoring samples, like for drug use. These drugs of abuse. So that's a common application for EMIT. And then in most of these cases, there are kits which are available. So it's a whole range of things that we can talk about, which may not fit in our lectures. But I will send you a link where you can get drug examples in drugs that you meet in everyday lectures. And these would be drugs that have a narrow therapeutic index. So we'll be talking of digoxin like we saw earlier. We'll talk of antidepressants. Okay, so there are all those examples that you can get from there. So I'll send you the link and you'll be able to see that. Okay, so let me go to fluoroimmunoassay. So you said that you have studied fluorimetry in other areas. So now we are just seeing that application in an immunoassay and these are fluorescence polarization fluoroimmunoassay and time-resolved fluoroimmunoassay. If we talk of the homogeneous type as an example, the antibody, the binding of the antibody causes some changes in fluorescence properties, such as polarization of the labeled analyte. And once this happens, the analyte concentration can then be monitored directly from the reaction mixture. So that gives, or that produces the fluorescence polarization immunoassay as a technique, because the binding affects the polarization of that of radiation. So in the early development of the technique, it was designed to work in a single tube, but development in technology has resulted in automation of this technique, and therefore can be used for screening assays, especially during drug development. The time-resolved fluoroimmunoassay, and this is a different technique because it can be adapted to either heterogeneous or homogeneous assay formats. This method uses chelates of lanthanides and palladium ions as the labels. So this particular method can either be heterogeneous or homogeneous, and the labels are the chelates that we had talked about earlier. So it has been used to test for drugs or hormones in biological fluids. This particular type of fluoroimmunoassays help to resolve or to cancel out fluorescence due to the presence of the biological matrix. And therefore, so this time resolved is capable of handling a biological assays without interference, from background fluorescence from biological materials. So the label is simply a chemiluminescent compound. Chemiluminescence is the emission of light resulting from certain chemical reactions. So from chem, which is chemical, luminescence, which is light. So these chemical reactions produce light photons as they occur. So these photons can then be detected using appropriate instrumentation. They can be detected and quantified because the amount of light that is produced will depend on the amounts of the product being formed. So in most cases, when a product is formed, it is going to be of high energy. And then as it goes to ground state, the excited electronic energy goes to ground state, and this is accompanied by release of photons. So in this case, we are talking of maybe an intermediate product, which is of high energy. And as it goes to ground state, then the light is emitted. Agents. The two which are commonly used agents are lumino and acridine ester. So the lumino and acridine esters are the most common labels that are used. Yes. Yes. Yeah, we started late. So is it a physical class or an online class? Have you received the link for the other class? Okay. So what I will do is I'll wind up at that point. Then I'll send you these slides. There was just one more slide I was to look at. So you look at it, and as part of the assignment, and we have headed the series of lectures for this unit. So have a good day. I'll also send you the link for where you can get drug examples that are analyzed by these methods. So look out for that in a short while. Have a good day.